synthesis. What are pyrimidines? Pyrimidines are aromatic heterocyclic compounds. These are nitrogenous bases. They have a molecular formula of C4H4N2. They are the monomers for nucleic acids. They have a 6-membered ring. They form nucleosides or nucleotides depending upon how many phosphate groups are attached to them through the deoxyribose or ribose sugar. Now, there are 3 types of nitrogen bases or pyrimidines in this case. These are uracil, thymine, and cytosine. Cytosine is present in both RNA and DNA. However, thymine is only present in DNA and uracil is only present in RNA. The main structural difference between cytosine and the other two is that at the 4th position carbon, cytosine has an extra amide group, which we'll see later is actually given by glutamine. Uracil and thymine also are structurally different because thymine is methylated or has an extra methyl group at the 5th position carbon, which is, as you can see, is absent in uracil. Also, we can see that the 2 nitrogens in the cyclic ring are present at positions 1 and 3. Carbons are present at 2, 4, 5, and 6. Okay, so let's go back to topic. Pyrimidine synthesis. Pyrimidine synthesis can be done by 2 pathways, salvage and de novo pathway, right? Salvage pathway, as the name suggests, salvage. Some sort of substances or molecules or some sort of substances that are present in the body that are not as useful as the product that is required. So they can be used to make through an easy pathway, added a few things to them, and they can make the product that is right now required, in our case, pyrimidines. De novo pathway is a pathway in which pyrimidines will be made from scratch. They'll be made from raw materials, they'll be made from base materials, which will then be built into the 6-membered ring and into the pyrimidine ultimately. So first we'll discuss de novo pathway. De novo pathway, as we said, it's from scratch. It's from base materials. In this case, there are three base materials or substances that are being used, right? And depending on what, in our case, the product is going to be uridine monophosphate. So the raw materials are carbon dioxide, and 2 of these 3 materials are amino acids. One being aspartate and the second glutamine. Carbon dioxide is being provided by bicarbonate ions. Comparing to the purine de novo pathway, the pyrimidine de novo pathway is very short. It's only got 6 steps compared to the 11 steps in purine synthesis, right? Now, another difference in the pathway is that the pyrimidine ring will first be made and then it will be attached to the ribose 5-phosphate. However, in the purine de novo pathway, the structure of the ring is made around the ribose 5-phosphate. And the product being formed is UMP, uridine monophosphate. There are 6 enzymes. Of the 6 enzymes, 5 are present in cytosol. The 4th enzyme, which is dehydroorotate dehydrogenase, is the only enzyme that is present inside the inner mitochondrial membrane. So now we'll discuss another point that we wanted to add was that which base product is providing what to the structure of the pyrimidine ring. So as we said earlier, that there are three base substances that will make up the product, which in our case is pyrimidine 6-membered ring. Carbon dioxide will provide the carbon at the second position. Nitrogen was present at two positions, at N1, at position number 1 and position number 3. The position number 3 nitrogen will be provided by glutamine amino acid, and aspartate will provide all the other, that is 3 more carbon atoms and 1 more nitrogen atom. These are present at positions 4, 5, 6 and 1, and you will have a pyrimidine ring. Okay. So the steps. There are 6 steps to the pyrimidine de novo pathway. The first step is the formation of carbamoyl phosphate, and we'll see the enzymes and whatever factors are adding to it. Then the step 2 is the condensation of aspartate with carbamoyl phosphate to form carbamoyl aspartate, and the reaction we'll see further. So carbamoyl aspartate is being formed. Step number 3, this is a very important reaction because cyclization, the closure of the ring, is taking place and you are being left with dihydroorotate. Step number 4 is where orotate is being formed from NAD dependent dehydrogenation or oxidation reaction. Step number 5 and 6 are very closely linked. So what happens is that ribose 5 moiety is being added to orotate, which forms OMP, and OMP is being decarboxylated. Uridine monophosphate is being decarboxylated to form our product, which is uridine monophosphate, and this is step 5 and 6. Okay, enzymes. What enzymes? There are 6 steps and there are 6 enzymes. So very important mnemonic for the enzymes, which is very helpful for remembering the enzymes, is CAD-DHO2. Now, by CAD-DHO2, it also helps you remember what the enzymes are another part of, right? So CAD is C is for carbamoyl phosphate synthetase 2. A is for aspartate transcarbamylase, and as I'm telling you these enzymes, these are according to the steps as well. So step number 1, carbamoyl phosphate synthetase 2. Step number 2, A, aspartate transcarbamylase. Step number 3, which is CAD, so D, we have dihydroorotate. Now CAD, which is carbamoyl phosphate synthetase 2, aspartate carbamylase and dihydroorotate are part of multifunctional enzymes, which I'll tell you later what really they are, right, and what are their advantages. CAD-DHO2. Now DH stands for the 4th enzyme which catalyzes the 4th reaction, dihydroorotate dehydrogenase. As stated earlier, this is the only enzyme which is present inside the inner mitochondrial membrane. Now, the only enzymes left are 2, which catalyzes 5th and 6th step reactions. So, O2. The first one being orotate phosphoribosyl transferase. The 6th one is uridine monophosphate decarboxylase. And these are part of bifunctional enzyme, which is UMP synthase. And as said, the mnemonic is CAD-DHO2. Let's look at the reactions now. The first reaction is between 2 of the 3 base substances that are taking part, or base reactants that are taking part in this reaction, and that is carbon dioxide and glutamine. Carbon dioxide and glutamine react under the enzyme carbamoyl phosphate synthetase 2 to form carbamoyl phosphate. In this reaction, 2 molecules of ATP and 1 molecule of water is being used to give off glutamate, 2 molecules of ADP and inorganic phosphate molecule. Now, carbamoyl phosphate in the second reaction will condense with aspartate under the enzyme aspartate transcarbamylase and form carbamoyl aspartate. Carbamoyl aspartate will form dihydroorotate by the enzyme dihydroorotate. This is not a matter of whether dihydroorotate is only being formed, it's a matter of cyclization that is actually occurring to complete this structure, this cyclic structure. And during this reaction, carbamoyl aspartate is being made into dihydroorotate along with the release of water. Now, dihydroorotate will be oxidized by a NAD dependent oxidation reaction by the help of enzyme dihydroorotate dehydrogenase into orotate, and obviously NAD will be reduced to form NADH and H positive. Orotate will now become orodyne monophosphate by the enzyme OPRT, which is a short form for orotate phosphoribosyl transferase. So in this reaction, ribose 5-phosphate moiety is being added to orotate to make it OMP, and the ribose 5-phosphate is being given off by PRPP, and a molecule of inorganic phosphate is being given off. Orodyne monophosphate is decarboxylized to finally form uridine monophosphate, and this is done by the enzyme OMP decarboxylase with the release of a carbon dioxide molecule. So this are the reactions. Now, we've only formed UMP by this stage, which is only one of the 3 pyrimidines that we have. So how do we form CTP? UMP is uridine monophosphate. Uridine monophosphate will be continuously or phosphorylated twice to give UDP and UTP successively. UMP is first by the enzyme kinase, which adds a phosphate group into the reactant to form the product UDP, which is uridine diphosphate. The phosphate is given by the ATP, which leaves as ADP. Again, phosphorylation of UDP, uridine diphosphate, takes place to uridine triphosphate. ATP gives the phosphate group by kinase. This reaction occurs. Now from UTP, CTP will be formed. CTP synthetase will act and form CTP. Now if we remember, CTP, the pyrimidine ring of cytosine, the nitrogenous base here, has an extra amide group on the 4th position carbon. That amide group is provided by glutamine, right? Glutamine reacts with a molecule of ATP and water under a CTP synthetase and forms CTP, glutamate, ADP, and inorganic phosphate is given off. Now the formation of thymidine monophosphate. Thymidine or thymine nucleotide or nucleoside is only present inside DNA. So it will not have ribose sugar, instead it will have deoxyribose sugar. Now here there's two ways into making thymidine monophosphate. The first one utilizes UDP. UDP will be converted into DUDP, that's deoxyuridine diphosphate, by the help of ribonucleotide reductase, which is using thyrodoxin to reduce this ribose moiety in the UDP. DUDP will be converted into DUMP by dephosphorylation, right? So you're removing a phosphate group and forming DUMP. If we remember it, our thymidine will have, the pyrimidine ring will have an extra methyl group on the fifth position carbon. This methyl will be provided by the methylene tetrahydrofolate by the enzyme of thymidylate synthetase DTMP, or thymidylate will be formed. Thymidine monophosphate, deoxythymidine monophosphate is formed, and tetrahydrofolate is demethylated. All right. The other reaction is we're starting off with both UDP and CDP, right? UDP and CDP by ribonucleotide reductase that we've already seen in the previous reaction using thyrodoxin will convert these into their deoxy forms. Once they're converted into their deoxy forms, now they will be phosphorylated by the enzyme kinase, nucleotide diphosphate kinase, and obviously ATP will give either both of them a phosphate group each. CTP, the deoxycytidine triphosphate, is converted into deoxyuridine triphosphate, which is then converted, which is most probably converted by a deamination reaction, because the only structural difference between both of them is that cytidine has cytosine pyrimidine ring has an extra amide group at the carbon position 4. This will then form DUMP, obviously by dephosphorylation, which is catalyzed by DUTPase, right? DUMP will now be converted to thymidine monophosphate, again by thymidylate synthetase. Okay. Now, what are multifunctional enzymes? Multifunctional enzymes are proteins that contain at least 2 distinct enzymatic activities and 2 distinct active sites. These are only one enzyme, and unlike complexes, do not have more than one. Their subunits are not distinct enzymes. Okay, so what are their advantages? Multifunctional enzymes, they increase the speed and efficiency of a pathway, and they decrease the concentration of intermediates, because obviously active site after active site is helping in successive reactions. So you're not letting any reactants sit around, you're not letting any intermediates sitting around, you're producing products and you're pushing them forward. So yes, regulation of de novo pathway. Carbamoyl phosphate synthetase is mainly a enzyme that is being regulated in this scenario. It is inhibited by feedback inhibition. So the end product of the reaction is going to act upon the enzyme which is catalyzing the first reaction, inhibiting it, that there's already enough product, you don't need to make more, stop. Feedback inhibition. UTP and UDP are inhibiting carbamoyl phosphate synthetase 2. Carboxyl phosphate synthetase 2 is activated by the enzymes ATP, not by the enzymes, by ATP and PRPP. Aspartate transcarbamylase is inhibited by CTP, and UMP and CMP are inhibiting uridine monophosphate decarboxylase. Okay. We've completed de novo pathway. Salvage pathway, as the name suggests, we've already discussed, is that salvage. You're salvaging as not important substances present in the body which can be used to make certain more important products, right? So there can be bases available, there can be many different molecules that can be added to and formed pyrimidines. So in the first case, uridine and cytosine. Uridine and cytosine will react with ATP and gain a phosphate group to form UMP and CMP by the enzyme uridine cytosine kinase. And ATP is already, released. Excuse me. Then deoxythymidine will again react with ATP under the enzyme to give off DTMP and ADP, which is thymidine kinase is the enzyme. Deoxycytidine again reacts with ATP by deoxycytidine kinase to form its monophosphate. Uracil, thymine, and orotate react with PRPP and gain a ribose 5 moiety to form UMP, DTMP, OMP, and pyrophosphate under the enzyme pyrimidine phosphate ribosyl transferase. Okay, that was a few ways that we can salvage other materials to form pyrimidines, right? So in another example, if you have cytosine and thymine lying around from DNA breakdown, right? So cytosine can be made into uracil, which will react with ribose 1 phosphate and uridine to cytosine will by deamination form uracil. Uracil reacts with ribose 1 phosphate and under uridine phosphorylase forms uridine and by nucleoside kinase forms UMP. Now thymine is also left over by any DNA degradation, or you don't need it for DNA formation anymore. So thymine will react with deoxy 1 phosphate. Under the enzyme thymidine phosphorylase, it forms thymidine, and thymidine then forms DTTMP. So this is it. Thank you so much.